I just want to introduce Mohammed Uddin. He's a postdoc over at SickKids. He did his undergrad and his PhD at Memorial University in human genetics, and then he was lucky, or we were lucky, to get him at the Center for Applied Genomics at SickKids to work in Steve Scherer's lab, and he's going to talk about an integrated genomic approach to identify genes associated with developmental delay. And thanks a lot for doing this for us, Mohammed.
Thank you, Ganesh. So I have 10 minutes. I'll be talking with a very speed pace because they will cut me off after 10 minutes.
Okay.
So just an introduction of the developmental delay. It's a collection of rare conditions and that primarily includes intellectual disabilities, autism spectrum disorder, mental retardation, and learning disabilities. And the population prevalence of developmental delay is about 3%. And the primary diagnostics for developmental delay patients are the chromosomal microarray, and it has a very good turnout, right? Because in developmental delay, we tend to get large deletion and duplications for about 10 to 15% of the cases. So even if you do next-gen sequencing, those large variants still there. So the clinical microarray is very low resolution because we are targeting to detect large CNVs. So it's 180K, but the probes are really good. They're validated probes. And we end up getting about 10 CNVs per sample. And usually, because it's clinical, it's performed in a clinical setting, so we don't usually have platform match controls. So it's just case data. And what the clinician does is for each of the patients, they look at the 10 CNVs and ascertain them whether the CNVs are whether clinically significant or some variant of uncertain significance because there is lack of evidence what the gene does for those variants, or if the variants are common in population, that's a benign CNV. So the problem with the clinically significant and VOUS variants are we don't know what are the genes that are associated with the death delay, even though we know the prevalence of these clinically significant variants are high in cases compared to controls, but these variants tend to have like 30, 40 genes, and we don't know what are the genes that really associated with the condition. So this is the data we have. It's coming from two different hospitals, SickKids Hospital and Prairie Valley. So the total discovery data set includes 10,000 samples, 10,600. And we also integrated other samples from other databases. So at the end, we have about 29,000 cases to analyze. And as I said, we don't have match controls, but we have high resolution microarray controls, and we have about 10,000 of these new controls for this study. And we also have downloaded and using this other 11,000 controls from other databases. So we have about 20,000 controls. So initially, anybody will think, oh, you have a case control data, why don't you do a case control association study? Well, you can do it, of course, but the problem is the control variants are small for the clinically significant variants. Even if you do gene level association, you'll end up getting so many genes to be with high p-values. So at the end, we still do not know what are the genes because you have one of the study in Nature Genetics came out showing 3,800 genes have a p-value at least 0.001. So really we don't know how to approach this to find out or tease out at least the candidate genes. So this is the problem that I'll be discussing for this talk. So this is the diagram of the whole data. So about 5% of the patient are diagnosed with a clinically significant loss. VOUS variance is about 18% for loss, and gain is about 1.8% gain or duplication are clinically significant, whereas 31% gain are VOUS, because for gain it's hard to determine the pathogenicity because we don't know where the other duplicated part of the region in the genome. So we did a screen of the known syndromic loci to see what are the syndromes in our 10,000 cases the enriched, just the known ones. So it looks like there's 1%, there's one region that reaches 1%, it is 22q11. deletion and duplication. Then there is well-known region like 16p11.2 that is about 0.8%. So it is a mix of different syndromes, this whole cohort. So this is the pipeline what we are doing and we will do in the next couple of days. So after getting the clinical annotation, we are doing three different analyses. One is called critical exon analysis. The second part is the protein expression network analysis for those genes. And for the non-coding part, we are doing enhancer enrichment. Initially, we just wanted to see if the clinically significant variants have any male to female bias. So we found that female are ascertained for more clinically significant variants compared to male, because female requires more pathogenic variant to manifest their phenotype compared to the male. But that's a completely different discussion. So this is what we found. We see this in the combined data, which is the top diagram. Also, we see the trend in individual analysis in the two hospitals. So in terms of number of genes in the variants of clinically significant and VUS, you can see the numbers of genes are in thousands. So it's really tough to know what are the genes. So the critical exon analysis that we initially use is, the definition of critical exon is if an exon has a low mutation burden in the population, but it has a high expression in the brain, so that exon is conserved for functional relevance into that tissue. So this is what we call critical exon. And this exon shown to be highly targeted for de novo mutation in autism spectrum disorder. So we already published this paper in Nature Genetics showing the enrichment of de novo mutation in critical exon. So what we wanted to see if the critical exons are enriched in clinically significant variants for certain genes compared to the control genes. So this is the results. Look like the red dots are showing the critical exon ratio for the clinically significant variants genes, and the blue dots are showing critical exon ratio for the control genes. So the top panel is for loss CS and loss VUS. The bottom panel is for actually anyways, the bottom panel is for gains. So you can see that there are statistically significant critical exon enrichment in gene sets with clinically significant variants. So next, having that as a tool, what we did next, we took the proteome expression data that came out in Nature, the human proteome map. This is Fourier transformation data from adult to prenatal to adult tissues. And we use 30 histological tissue from this database to construct a proteomic map. So we applied WDCNA to cluster the genes, and we obtained about 23 cluster of genes. And then for each cluster, we went through the gene ontology database to see what each cluster means. Then we found one cluster, or module, we can say, is really enriched with synaptogenesis and brain development. And then we wanted to see if that module is also enriched with critical exon or not. Then it turns out the panel, D panel, showed the critical exon enrichment of that module with the empirical p-value. Then this four permutation distribution shows the enrichment of clinically significant and VUS genes in that module. So then what we did, we combined the critical exon and the proteomic map analysis to come up with candidate genes. So this is the criteria, primary criteria that we use. So the gene have to be highly enriched highly with critical exon in human developmental brain, and it must be part of the blue module that I showed in the earlier slide. And the gene also have to be impacted by one CSR VUS variant. So then these genes are our candidate genes to follow up with more genetic data. So this is one of the variant we are following up. So it has multiple de novo variants. detected in MSH6 and FBXO11 gene. If you look at the gene, the mismatch repair gene already been associated with cancer, but it is a low penetrant gene. But the FBXO11 gene is, we followed up this gene and we found additional 3 de novo deletion in other databases. And also the exome sequencing showed 1 de novo SNVs in an autism program. So we think the FBXO11 gene is a very good candidate gene. Then we looked at the protein network and we saw the first degree neighbors, and we saw the red networks. The red genes are the genes that have a de novo mutation from exome studies found in autism program, that is highly connected with the FBXO11 gene. So these genes, the protein module, the protein expression is similar, behaving similarly with the other candidate gene for autism. So, and this gene already been implicated with otitis media. Otitis media, it's a family-based association analysis for, it's a type of hearing loss, and also knockout homologs in mouse showed it resulting in deaf mutant. So it can be a very good candidate for developmental phenotype. Another gene is the PPP1R9A gene, that is a maternally imprinted gene. And this gene, we have the shortest deletion in this gene in our cohort that is de novo, not found in parents, but in the proband. And following this gene, we also found 2 additional de novo deletions, and 1 de novo SNV is reported to be in an autism program. So this gene also maternally imprinted, and it is biallically expressed in epithelial tissue, but for the CNS tissue, it is maternally expressed. And the protein encoded by this gene is involved in neurite formation. So I'll end up the talk by acknowledging my supervisor, Stephen Scherer, James Serapoulos, and Marsha Piva, who also involved providing the data, and all other statisticians and bioinformatics people who helped us on the analysis, and different databases where people helped us on following the mutations. Thank you.
Okay, any questions?
Yes.
I have a quick one for BrainSpan. I believe there's RNA, there's sequencing data, and there's also exon array. Does the critical exon stuff, I always wondered, does that work for the exon array measurements as well?
Yes, we did the critical exon we did the critical exon measurement using RNA-seq and microarray data. So in our Nature Genetics paper, we use both data, and they both kind of shows robust results for critical exon. There are some exon in the gene that are really showing high expression in both platforms and do not have any mutation accumulation in general populations. So it's really interesting to see some of these exons are really preserved for brain function and brain regulation. Yeah.
Any more questions?
I guess.
Yes.
Is the protein data quantitative or qualitative?
It's quantitative.
Okay.
So it's
I'm thinking of a different paper.
No, it's like for each gene is the average spectra count.
Okay.
Quantitative data.
Thanks.
So I wonder if these copy number aberrations, alterations, are they de novo or are they inherited from parents?
Okay, so I haven't showed the whole data. So we have about 160 de novo mutation in the whole cohort. But usually when in the clinic, when the patient comes in, they really do not check for inheritance all the time, because most of the time the parent gene is not available. But you can look at the variant. If it is 16p11.2, you already know it's a pathogenic variant, because you don't see it in normal population at all. So even though the pathogenic variants are there, we don't know the exact estimation for de novo, just because the nature of the clinical data is like that. But if it is a research data, that would have known their data.